All right, let's get started, everybody. It's an absolute pleasure to have Doug Shepard here with us today. I've known Doug for about 10 years. He's a great guy. Currently, he's at Arizona State University at the Center for Biological Physics and the Department of Biological Physics and the Department of Physics. Prior to joining ASU, he was at the University of Colorado, Denver. He earned a PhD from Colorado State University and an undergraduate degree from the University of Santa Barbara, University of California, Santa Barbara. His focus his research focuses on developing quantitative imaging and statistical inference tools to build predictive models of genetic regulation in multicellular systems. Something interesting about Doug that some of you may not know is that for quite a while there, Doug was a sponsored athlete doing ice climbing all over the world and has many first ascents, including in Alaska and other places. So with that, I'll give you the floor, Doug, and I'm very interested to hear what you have to tell us.
Great. Thanks, Ryan, for the introduction and bringing up my other life, which I don't always talk about too much. And so, you know, what I want to focus on today is some efforts that we've made since I moved to Arizona, which is just about 2 years ago, a little more than now, on really, in my opinion, trying to push these imaging techniques that we use in biophysics into dynamical systems at the nanoscale. And so I'm going to focus a little bit less on the biological findings and the molecular labeling portions of my group today. And I'm going to give a little bit more of kind of a physics of optics and how we can exploit the wave nature of light to improve the way that we take data in 3 dimensions. And so I think, you know, hopefully there's going to be a bit of review. Hopefully it's useful and helpful. And so, you know, we'll go from there. So what you're seeing here is a multicolor image of a fixed piece of tissue. And in this case, it happens to be lung tissue isolated from a rat. And you see different cell types and different structure labeled. And so this is generated on a type of what's called a light sheet microscope. And so I'll actually show the exact microscope that generated it. The important thing here is we have, you know, sort of subcellular nanoscale resolution, and we can acquire this image in a matter of minutes. And so what we're really trying to do is push things forward on that scale so we can come back around to the modeling side to think about, you know, what's happening. So what really drives a lot of our interest in doing these, you know, high-speed imaging experiments where we're trying to really be quantitative. And what I mean by quantitative is down to like how many photons arrive level and how confident are we in that, is this idea that I got really interested in as a postdoc. As a graduate student, I worked on semiconductor quantum dots, and I looked at the interactions that 2 quantum dots might have with each other. And then when I was at Los Alamos as a postdoc, I started working on biological systems and this idea of sort of stochastic decision making. But I think that's a bit of a simplistic way to view it sometimes. In reality, some evolutionary pressure has caused these systems to go from where they have an input, and then, you know, maybe they're always going to have the same response to that input. And so, you know, I would call that deterministic, right? So maybe we have 2 dimensions of an input, heat and salt concentration, or heat and nutrient availability. And the regulatory system is primed to always do the same thing. You could have diversity generation where the one input generates a lot of responses. So you could imagine, you know, this might be some example of bet hedging, right? So the population is going to generate a wide variety of responses because evolutionary, this is found to be advantageous for the population to survive. Or you might have buffering where a wide variety of inputs all lead to the same output. So here it could be evolutionary. If any of these is seen, the population has to respond the exact same way, otherwise the fitness goes down. And I really try to think about it in this way instead of anthropomorphizing the system and saying it's making decisions, and that these are chemical reaction networks that have evolved over time to act in a certain way. So if we want to probe those, there's a lot of availability nowadays, and there's sequencing, there's you know, standard biochemistry experiments. Optical imaging has become a really, combined with molecular labeling, has become a, you know, really prominent way to deal with this. And so this is an example of a plot from a piece of work by Arjun Raj, who invented a sort of modern method of RNA fluorescence in situ hybridization. This is based on work that dates back a long time, but particularly his work evolved the work by Femino and Singer. And what they've done here is they've just measured the housekeeping gene GAPDH They do this in such a way they can attach fluorescent tags to individual RNA transcripts, and then they can count how many RNA they are per cell by taking a picture and localizing the spots. And what they found was that if they looked at, say, cell cycle, so here, cell cycle is in blue, green, and sort of, I'm sorry, blue, purple, and sort of lighter purple. They found there's not really a lot of correlation of the cell cycle of the cell. So if it's dividing or it's, sort of doing its wherever state it happens to be in. But in fact, there's almost a direct linear correlation with the volume. So this would be an example of doing an extra quantitative measurement and realizing that what appeared to be stochastic or variable outputs at first actually are well explained by having this other hidden input that you would have missed with another type of measurement. And so to me, this is an argument for trying to build fast multidimensional measurement tools and making sure we're very confident in them, that maybe we need to just be measuring some more pieces to really understand the way that these systems are working. So you could ask, well, what really leads to that diversity generation? So here's a beautiful movie from my colleague, Tim Stazovich at Colorado State University. Brian Munsky probably showed some of his data. And what's actually done here is Tim has actually created a genetic reporter to watch RNA and new protein synthesis as things float around inside the cell. So you can see little dots floating together. Sometimes you can see two colors together. So Tim's actually able to watch protein being made. And so you could just say, well, if we measure all of these, we might just find out the only variability is whether or not two molecules run into each other. This is work that actually I've done with Brian, where we looked at this idea of population bet hedging. This was also in collaboration with Gregor Neuert. And here we use that same RNA fish method. We coupled this with some fairly advanced modeling ideas to come up with, can we understand from snapshot data how a system might respond to an external stimuli? So in this case, these are yeast. They're responding to a salt shock. And what these green and red puncta are individual RNA that are being made. And the important thing about this method is we have to kill the cells. So this is like a flipbook movie. So in each of these, we need to image lots and lots of cells because we need to get a probability distribution so that we can actually do the modeling. Turns out if you put this all together correctly, you can actually create predictive models of the way the population is going to respond. You don't know the way any given cell is going to respond, but you can do the population. To do that, you need a lot of data. So again, it's this argument for really going fast, really being able to churn through a lot of cell imaging really quickly. And finally, I'll just come back to Arjun's example for this sort of brief biological motivation of they then went and measured a whole bunch of RNA. And what they found is not every type of RNA is correlated to the volume of the cell. And so this argues that there are other hidden parameters there that even when you can successfully do this type of imaging, platform-based experiment where you're literally counting the number of RNA inside every single cell, that's still maybe not enough. You need to include some other information in your experiment. So if I was to sum up the kind of ideas that you might see in this area, you would say, well, I'm going to take some RNA fish data. This is what my group would do. So over here we have RNA in green and orange, just sort of these nice, bright colors. Each dot's an RNA. We're going to try and pair that with model identification, pair it with some computational image analysis, attempt to down select to the right model, fit and predict our data, and go through this whole loop. So as we push further and further into the nanoscale, what is often lost in this type of feedback loop between computation and theory and experiment is the actual response of the instrument and a real quantitative understanding of the way that the instrument is collecting the photons from the sample. So why is that important? So let's take this illustrative example of something that has been enabled by new microscopy. So in 1984, there's this nice paper about the fact that microtubules, which are part of the scaffolding of the cell, they have this dynamic instability. And so there were some observations of this in vitro. Then about 10 years later, there was this very nice paper, Deborah Figerson was the first author, where they actually saw this somewhat stochastic behavior where this microtubule tip would start going out its origin, then it would collapse a little bit, then it would go this way, but then all of a sudden the entire tubule would fall apart. And it turns out they could fit this to a pretty standard stochastic model of growth. So as new microscopy and fluorescence labeling methods have become available, it turns out you can actually go into the cell and do this experiment. So this is a paper from a few years ago from Jennifer Lippincroft-Schwartz lab. We're actually combining a new type of microscopy, structural illumination, that I'm going to talk about in the rest of this talk, and some genetic fluorescent labeling tools. They're actually able to observe this dynamic instability in vivo. I don't want to say in vivo because it's not in the animal, but it's in a live cell on a cover slip instead of having isolated microtubules. And so the only way that this type of measurement to see that what we actually saw for an isolated molecule on a cover slip actually translates to inside the cell is to have these kind of simultaneous advancements in imaging, and molecular labeling. And then that provides pretty rich amount of data. So I want to give you two motivating examples of things we're interested in the lab at the moment. And I'm going to flat out say we haven't solved both of these problems, but these are the motivations for the microscope approaches I'm going to talk about for the rest. So in a project with Nick Galati, who's at Western Washington University, we're trying to understand how proteins move in cilia. So if you're not familiar with cilia, I'll play this movie. And cilia are the fine hair-like structures that are on the outside of this cell, and they're whipping back and forth. And it turns out they have a power stroke phase in which they're pretty rigid. And then they have this return phase in which they go quite floppy and then go back before they do a power stroke again. What's amazing is inside of those cilia, actually protein are transported. And so one of the questions is, how does that work? How does this regulate how the cilia are interacting with their environment? And what I want to tell you is that these cilia are oscillating at something on the level of, let's say, 60 to 120 hertz. And they're moving, hopefully you can appreciate, slightly out of the frame of the microscope. So they're actually moving in 3D as well. So really, if we want to understand this problem from a microscope perspective, we need a technology, right? We need to be able to collect enough photons fast enough that we can both tell where the proteins might be inside of it, so maybe a fluorescent label, and how the cilia itself is moving. Another example is here at ASU, collaborating with Rizal Haradi's lab. He has a graduate student, Frankie, who's working on a real fundamental question in biophysics, which is, how do molecules move and propel themselves at low Reynolds number? So the drawing and cartoon I have here is from a famous paper by Purcell, which is called Life at Low Reynolds Number. And the idea is that if you have this helical structure, there's going to be some interaction with the fluid, and you're in a certain regime where you have sort of inertia versus what's going on with the fluid is acting in a certain way, such that you get these really complex movements of this flagella. And so here's a flagella that's been decorated with fluorescent dye, and this is just a 2D movie. And actually, the hard part about this is actually a corkscrew-like structure, and the rate of that precession that it has along this axis. So in this case, what do we have here? Capital omega is actually something you need to measure to understand the interactions with the fluid bath. So here we would need to be able to image extremely fast, continuous shaped objects that are nanoscale in 3D. And importantly, we need the resolution to actually resolve that term. So what I'm going to spend the rest of this talk talking about is two efforts that we've had to try and move towards resolving the kind of physics that's going on, going on in these problems. So to do that, I think a lot of people recently have learned about these methods known as super resolution. So the question is, should we just be considering doing one of these super resolution methods, right? And so the Nobel Prize was given to Eric Betsit, Stefan Hell, and W.E. Morner in 2014 for this class of methods. There's many other scientists, obviously, who've contributed to it. And generally, the idea of super resolution is you're going to try and increase the resolution of the microscope by introducing some sort of nonlinearity or sparsity. So what do I mean by that? So let's say we're interested in these two filaments. Let's not do a curved filament at the moment. Let's just take two straight filaments that are intersecting. So if we put this under a standard microscope, we're going to get a blurry image. So the microscope can't produce, infinitely sharp images. And I'm going to go into details about why that is after we talk about super resolution. So, but really, you have some ultimate resolution limit, which is set by the wave nature of light and the optics. That's going to produce these blurry things. And we can't really tell that much about the fact that there's two of these because they're blurred together. So these three main approaches that really were what won the Nobel Prize for, all attempt somehow to introduce a nonlinearity so that you can generate a sparsity so you can measure these two structures with very little background. So instead, a beam is structured into a donut. That donut then makes all of the molecular reporters, which in this case are fluorescent molecules, undergo stimulated emission, so they're not seen at the same wavelength as the middle of the donut under where you have normal emission. You then have to raster that donut beam plus the excitation beam around, and you would end up with this kind of image. So what I want to emphasize is it's a scan. And so this scan inherently is slow, maybe compared to the time dynamics you're interested in. For photo switching sim is very similar to STED. It's just that you have fluorescent molecules which can switch themselves on and off based on the photokinetics of the molecule and the light they're exposed to. It's slightly different than doing stimulated emission. But in the end, you still have to do some scanning. You still have to repeat a bunch of patterns. And so this is relatively slow. And then there's this whole class of methods that fall under single molecule localization microscopy. Often you hear the acronym STORM and PALM. Here you turn off all of the fluorescent reporters in the system, and then you randomly turn them on one by one. So if you can appreciate, in all of these, there's a nonlinearity. Instead, you actually literally have a step function where the donut occurs. In photo switching sim, you have many step functions. And in Storm Palm, you have a transition from off to on, which is an infinitely steep transition. So to introduce sparsity, all of these exploit nonlinearity. And so for us, none of these methods are fast enough is what it comes down to. It takes, hundreds of milliseconds to seconds to build images with these. And so while they have the resolution, they don't necessarily have the speed. They're also quite subject to background, so they're hard to work with in tissue. Okay, well, maybe we should just turn to the other sort of hot topic at the moment, which is deep learning. So there's a huge amount of papers in this area. And so the idea is you take some training data, you generate, some convolutional network, and then you say, okay, great, I trained it on one of these super resolution methods, and I can take my blurry picture because I had a low resolution instrument, turn it into a high picture, right? So here's our cell. We can turn it into this nice resolved picture. I really like this review, which is the lead author on this review is David Hoffman, where you have to be very careful, because if you have anything that looks like your training data, it is going to upscale that. So here now our cat, instead of just the orange patch on the cat, now it's, our network has assumed that might be the nucleus of a cell. And in truth, this is actually true. This isn't just a flippant statement. If you take ground truths that are squares, circles, rectangles, or triangles, you then sparsely sample those so they're slightly noisy. And then you train the network on only squares, only on circles, or only on rectangles, you get back only squares, only circles, or only rectangles. And so, deep learning has a lot of promise. And I think where it's really promising is where you run into a situation where you need to incorporate some sort of physics into the learning, right? So if you have a very difficult to evaluate physics model, you can teach a convolutional network what the output should be based on the physics of the system. But when you're just training it on images, none of that physics is included, right? There's no understanding for the system why you should get a square or a circle. It just is trained that it knows it should get a square or a circle. So let's back way up and just think from first principles, what do we need to do to make these measurements, right? And again, remember, we want to go really fast. We need to be at the nanoscale. So just to remind everybody, light is a wave. If light is incident on some sort of grating, we're going to get light diffraction orders. Those diffraction orders are going to come off at some angle alpha relative to the normal of the surface. Let's assume we have an infinitely large plane wave coming into this, and we have some spacing d. And then the other thing, we could think about light doing is, instead of interfering it with a grating, we can imagine it's going to go into some sort of media that has a different refractive index. And so there, the light's going to bend, and it's also going to change its relative velocity, right? So the refractive index is actually telling us something about the speed at which the wave propagates in that media. And so here we're going to have, some other process that's going to occur. So the two of these combined are really the main things we need to worry about when we're thinking about how light's going to travel through a system. And so when you put the two of those together, you can say, all right, well, I'm going to have my optical wavefront. For those of you that remember your basic optics, one way you can think about an optical wavefront is you decompose it into a series of Huygens wavelets, where each of those wavelets is emitting a spherical wave. You can also do this in the reciprocal space into sort of Fourier optics, but somehow you have to propagate that forward. And then you think about what happens when I put it through a microscope. So the simple optical imaging system I can think of is we have two lenses, right? So we're going to have some object that's at the focus of our first lens. We could decompose all of the light that's diffracting off that object into a set of Huygens wavelets. We're then going to collect that light with a lens. Any light that's at the focus will come out collimated. This is the rule of how lenses work. It's going to go into a second lens, and we're going to make an image. But you can also think about this in terms of the way that Abe did, which is when we think about the diffraction orders. And so here we put our diffraction grating, and we're going to get an image of our diffraction grating. So let's look at this sort of framework a little bit more carefully. So we have our diffraction grating. It's going to satisfy the Bragg condition, which says we're going to have some angle that's going to have to do with the number and the wavelength. We then are going to put this through an imaging system. We need the lens to satisfy what's called the von Buren condition. This just has to do with how the distribution of frequencies shows up here. So what I want to remind everyone of is that when you have an object at the front focal length of a lens, what's in the back focal length is the Fourier transform of that object. So for diffraction grating, we would expect a discrete set of peaks here. And the lowest frequency peak would be closest to the optical axis, and then the high frequency peaks would appear as we move further away from the optical axis. We then do another Fourier transform, the inverse transform, to get the image of our object. And ultimately, it turns out you can't keep increasing the resolution because as this alpha n keeps increasing, this is going to eventually not be captured by the lens. So how does that alpha n increase? Well, we can just look at this equation. If we keep shrinking d, then what's going to happen is these alpha n, we're going to get diffraction orders that are further and further spaced out, and eventually we're going to miss the first order. And if we miss the first order, we have no information about how that grading is spaced. So actually, this is where Abe's sort of resolution criteria comes from. And what this tells you actually is directly the definition of the numerical aperture of a microscope. It's the refractive index times the sine of the maximum collection angle. Sorry, the half angle. So the practical result of this is if we put a delta function perfectal spherical wave emitter here, what we end up is a blurred object, which is called the point spread function of the microscope. This directly characterizes the way the microscope works. Turns out we still have to make a digital image of that. So let's say we have two of these spherical emitters near each other. They're then really close to each other and we want to resolve them. Well, if we use a camera, an imaging system where the effective pixels on the microscope are pretty big, then actually we're only going to see one bright pixel. We can make them a little bit smaller. Well, it still kind of looks like one object. So we can hit the Goldilocks condition, which is actually called the Nyquist criterion. And here we can actually see that we've got two objects that show up on the digital detector. And we can go even further, but actually we start to lose. And the reason we start to lose is that there's only so many photons emitted by molecular reporters. And cameras themselves are noisy. And so what it starts to happen is we start to ruin our ability to tell this signal out from the noise eventually. So what I've just described to you is the traditional view of resolution that often is cited when we're thinking about super resolution microscopy or single molecule imaging. However, images consist of a broad variety of spatial features. They're not all one point emitter or they're not all a regular diffraction grating. So we could imagine that I have an object which actually is like a diffraction grating, except the frequency changes as we go across it. So the other effect of this microscope not transmitting all the spatial frequencies the same is that actually the image of this starts to get weaker and weaker as we go to higher spatial frequencies, or the contrast drops. This can be summarized in a plot called the modulation transfer function, where how much contrast we expect as a function of spatial frequency drops off. And in fact, you can have two microscopes that would perform single molecule imaging exactly the same, but will form entirely different continuous images. So what does that mean? It means their limiting resolution is the same, but the transfer function is different. And this is the key thing for the rest of this talk, is this transfer function. And just to really hammer that home, here's two different optical trains that have the same ultimate resolution out here at, again, this is spatial frequency, remember, so it's the inverse of resolution. But you can see this one has a very different transfer function. It drops to 0 and then bumps back up. If we take the inverse Fourier transform of this, we get the point spread function. So here we can see this nice airy disk. Here we see something where the center looks the same, but we have some rings around it. But here's what an image would look like for that. So out at these larger objects that looks about the same, as we go to the middle, we lose almost all contrast. But then when we come back to this inner ring, we actually have about the same amount of contrast, because again, remember, we have the ultimate same resolution. So if we want to make very, very fast 3D imaging, we need to generate images that have high contrast across the entire image. We also need to think about all the rest of the components. And so the detector read noise can affect this. So if we have one camera which has very low read noise, maybe we have some photon distribution in the end. Whereas if we have much noisier, we end up with a different photon distribution. So how to summarize this kind of very long and I know introduction, but I think it's really important to put everyone on the same grounding. is what we're interested in is going from point spread functions and transfer functions that look like this to point spread functions and transfer functions that look like this, where we have lots of contrast available in each of them. So in the kind of remaining half an hour here, I'm going to tell you about two approaches we've taken for that. One of them is to engineer the excitation light. So I'm just showing you a movie of what we're doing. And here you can see there's some diffraction orders. So we're probably doing something like diffraction. And the other one is we're going to engineer the collected fluorescence light. And so here we're doing what's actually called light sheet microscopy, but only with one objective. So actually what's going to happen here is we're going to spin this thing around and we're actually collecting very high quality 3D data, but just with that one objective very rapidly. So the entire first sort of little vignette I'm going to tell you is done by a postdoc in my lab, Peter Brown. And then the second part was work that was done in heavy collaboration with Andrew York at Calico Labs and Reto Fiocco at UT Southwestern. Okay, so how do we exploit the excitation light to improve the transfer function? So what we're going to talk about is two classes, mainly this idea that we can generate super resolution images by exploiting the way that we shine light on the sample. And I'll very briefly mention the idea of doing phase imaging. So the idea is we're going to take our light and instead of passing one laser beam in, we're going to pass two to generate structured illumination. So literally, we're just going to interfere the two beams. When you impose that interference pattern on a sample, let's say a fluorescence sample, you're going to get what's called a moire pattern. So here's a real life example of what a moire pattern looks like. And I'll pause it where it becomes real evident. This is a calibration test target. And hopefully here you can see on this pattern versus this pattern, there's a slight shift in the intensity profile. And we go to another one. And so what's happening here is different patterns are being applied to this fluorescent target. And so that moire pattern, what's actually happening is high spatial frequency features are getting mixed down to where the microscope can detect them. So this actually isn't a new method. This was really pioneered by Rainer Heinzmann and Mats Gustafsson. Originally, it was done with a diffraction grating. So they would just physically move a grating to create the patterns they needed. More recently, it's done with a liquid crystal and silicon spatial light modulator. And both of these methods really offer very high modulation contrast, and we'll come back to why that's important in a second. But they're both a little bit slow. Here we have to physically move something. Here, the physics or the engineering of how this works means that it can't refresh that fast. So to get high contrast, what you need is you need two laser beams that are going to come in. They need to have the exact same intensity and they need to have their polarizations be controlled. And in fact, what you really need them to be is you need them in this picture to be, let's say, S polarized, the way I've drawn this, versus P polarized. Because I also get a cross term when you actually do the vectorial diffraction theory that if I have this P polarized light situation here, I don't get as deep of an interference pattern. So this is just a bit, hopefully back to some basic physics of the way, almost a double slit works. But what matters here is that, again, if you don't take anything else away, the intensities have to be balanced and we need to maintain the polarization. So one way we can try and make these systems run faster is we can use what's called a digital micromirror device. And so what a digital micromirror device is these two little mirrors, and there's about 1 million mirrors on this chip. And what they can do is they can tilt. And they can tilt to an on or an off position. And so people realize this, and they've actually done structured illumination with one wavelength using these. So here's a list of sort of papers that have done one wavelength. So why have they only done one wavelength? Well, turns out when you have a grating like this, you both get the diffraction grating that has to do with the pattern that you put on there, but also the shape of these mirrors imparts a sinusoidal envelope onto all of the orders of your diffraction grating. And so even if you pick two orders that you think will give you what you need to interfere correctly, they may not have the same intensity. And so this was a problem that hadn't been solved because it turns out it's a really tricky thing to think about. How do you correctly geometrically arrange all of this so you get perfect interference, which again, remember, is that polarization and intensity are the same. So Peter Brown in my lab thought about this really carefully. And what he realized is there is an analytical solution to this problem. And so I'm not going to go through all of this theory. What I can say is it has to do with taking these ideas in the far field and then treating the DMD as sort of a lattice and then summing over unit cells. But the details are on the supplement of his paper from this year, which the citation is here. And all of his code for the simulation is open source and is found here. It's all in Python. So what this allows you to do is generate these plots. So what am we plotting here? Anything in the solid line is the on mirror direction. So when the mirror, let's say, is facing this way. Anything in the dashed line is when the mirror might be flipped to the other direction. And these are telling you areas where the blaze condition is satisfied, which means the diffraction order you want is lined up with that sinusoidal envelope of the intensity that goes on top of it. So he simulated this for 4 colors, and it turns out, well, there are configurations of the incoming angles of the light and the angle of the DMD relative to the outgoing optical axis, where you have both 3 and 4 color solutions. And again, he was analytically able to do this, which means this calculation takes seconds. It also turns out there's a 3-color in-plane compromise where your DMD doesn't have to be so heavily tilted with respect to the optical table. And this is what we end up using. So you can see the green dash here is slightly off from the blue and the red. So I'm going to show you what that does in a second. What happens is that sinusoidal envelope ends up being slightly off from the diffraction order. So this is the real world consequence of not lining the system up correctly. This turns out to reduce our modulation contrast by about 2%, but it makes the alignment significantly easier. So with this model, Peter's then able to then exactly model the way that patterns should then show up as diffracted orders off of the DMD device. And the nice thing about this, if remember, we're talking about diffraction gratings, we know that the way light's going to diffract is dependent on the wavelength. So what we can do is we can actually create unique patterns for each of the wavelengths we want to use. And then we can actually look at the diffracted orders that come off of that and pick all of the ones that are overlapping. So in this case, that's going to be these yellow dots here are the ones that we want to use for our system, because they're all going to have roughly the same spacing of interfered beam, which means even though we change the wavelength, we have the same spacing of our sine wave for the interference. And they all have roughly the same intensity, which means we get a high modulation contrast. Really importantly, because he has a very streamlined analytical model, we can actually use this for inverse modeling as well. And I'm going to talk about that in a second. And this allows us to really understand the way the system works. So you can validate this model by just making a theory prediction of where are all the diffracted orders going to show up, and what is the intensity of all of those diffracted orders going to be. So the heat map here tells you what the intensity should be for every single diffraction order, which these are all actually tiny little dots for all sorts of different patterns applied to the DMD. So this is about 3,000 different patterns that he used to do this. You can then just put a camera at the Fourier plane for a lens looking at our DMD device, which is here, and just record the intensity for each of those patterns. And I hope everyone can appreciate that there's almost exact overlap between these two, which means we can actually now use this model to tell us exactly how to create a polychromatic light modulator that's capable of running tens of thousand frames per second. So we're now 2 orders of magnitude faster than any previous modulator that's been used in a structured illumination instrument. We can also use this to directly measure that transfer function that I spent so much time carefully explaining. So let me walk you through how that works. So we know exactly how those peaks are going to show up at this Fourier plane here. We then are going to conjugate that to the back focal plane of the lens, which is also often the pupil, the way these objectives are built. We'll then focus that pattern down onto a thin dye slide, and we'll record the fluorescence that occurs. So here we know our diffraction orders. We only allow these 3 through. We do that by putting a physical mask there, and we block the rest of them. It's actually a piece of foil that we poke holes in with a 3D printed template. That then shows up as, a sine wave with some modulation on here. That sine wave has some resulting, Fourier transform, which is going to tell us about the patterns that went in, the frequencies that went into making it. We then record the irradiance that occurs from this, and that ultimately tells us the entire transfer function of the system without ever having to measure the point spread function. So actually, we can do this with much higher fidelity than is normally done, because again, we can just put lots and lots of patterns on and then check to see how well we did. And so from that, we can calculate this modulation transfer function. And this is what the data looks like. So you can actually, here's our fluorescence slide. You can see there's these fine grained patterns and then an overall larger pattern. What we then do is compare it to our model for how much contrast should we expect at each pattern. And what actually we find is that, because we have a real optical imaging system with noise and no optics are perfect, that we deviate from, the ideal theory of the way that the objective we're using is fairly significantly in certain areas where we have less contrast available to us. A point I want to make here is this is always true. It is almost impossible to get a high numerical aperture microscope objective that meets theory. So how do we then use this to increase the transfer function? So what we do is we apply that pattern to some fluorescence sample. In frequency space, remember, we can only collect some maximum angle. So if this is Kx and this is Ky, so our spatial frequency in X and Y, we can only collect out to some maximum. We're going to see some discrete peaks due to our pattern. What we want to do is we want to figure out what data was downmixed in that Mori pattern and shift it back out to higher frequency. So what does this actually look like? So here's a fluorescently labeled cell where the actin cytoskeleton is labeled with a dye molecule. The pattern we're applying is in this orientation. And here's the Fourier transform of this. So you can see here's the peaks that correspond to the pattern. Here's that extra data that we're interested in understanding how to unwrap and somehow use that information to not go from the situation where we have this image where the pattern is convolved into our image, but actually we remove the pattern and we gain the high frequency information. So to do that, we have to take data at a bunch of angles. And then what we have to do is solve this inverse problem. So the inverse problem is as follows. We have our true object, and that's multiplied by our pattern, which is described by this 1 plus a cosine. That is then convolved with the point spread function of the imaging system. And then there's noise applied. And so what we have to do is invert this whole thing. And anytime you try and invert an equation and you're subject to heavy noise, you have to be very careful about how you do this to not invent things because it's hard to regularize. So here's just a quick simulation to show you kind of what can happen with that. Here's the ground truth of some small filaments. Here's what the normal microscope would see. Here's the sim reconstruction where we invert this equation. And then what we did is we took that ideal case where we think we have more signal to noise than we actually do for our real transfer function. And we just asked, what's the result for the real case versus the ideal case? And actually you can see here with the real case that we actually recover the underlying signals, particularly between these two overlapping filaments, which is here, better than we do for the ideal case. So that's at low signal to noise. If we just increase the effective brightness in the simulation by raising the signal to noise, we start to see that it doesn't matter so much. But remember, we want to go really fast, which means more likely we're not going to collect so many photons. So it turns out that you just have to be really careful about how you do this inversion. And eventually what you end up with is this beautiful image in Fourier space where we've reassigned all of that information in the Mori patterns to their correct areas. You can actually see the areas that are contributed to by each angle. You can actually see one of our angles has more contrast in this image that I picked than in another one. And then what we have is an effectively much higher resolution. So this also is open sourced, and we've compared our way of doing it and doing this all correctly to a ground truth or a really nice package that people sort of take as the ground truth in the field called FAIRSIM, which is written in Java by Marcel Mueller and Thomas Huser. And what we find is that our approach works almost exactly the same as theirs, so we're really confident in it. And so here's just some results from it. So we can get this specially made calibration slide from this company Argolite, where they etch these patterns into this glass and then they fill them with dye. And one of the best patterns for sim is they actually have these two lines that are spaced close together, and the lines get 30 nanometers closer every line pair. So here would be the normal microscope. We can't even resolve 180 nanometer spaced together line. In our sim, we can go down to about, let's say 120, maybe 90. If you use some of those deep learning ideas that I mentioned we were a little bit skeptical about, but you apply them to our data, you can see you start to recover something, more around 90, a bit clearer, but you also have this weird pixelation going on. So really we have some effective resolution, let's say between 90 and 120 nanometers, which is about 1.7 times the diffraction limit for this system. So we should actually, normally we would only have, let's say, something on the order of like 280 nanometer resolution. We can image just say fluorescent collagen on a slide to see how we do. So here we can actually see we resolve these individual collagen fibers. You can actually see a little bit of reconstruction noise in this image. And then what we can get to is now we've done sort of the largest field of view to date in one of the faster kind of live multicolor sim experiments. And this is just an example of that. So what we're tracking here are mitochondria in cyan, lysosomes in pink, and then the underlying actin cytoskeleton in kind of this greenish color. And the thing I want to point out is actually it's a bit blurry here. So the reason it's blurry there is because of this out of focus fluorescence. So it turns out sim is still really subject to light hitting it from above. And so even though we've improved the transfer function laterally, we really haven't done much axially. And so, this is the next step to be able to do those types of experiments we're interested in is, how do we get better axial resolution? So for us, what we're really thinking about doing is instead of using fluorescent emitters, we're going to try and do refractive index imaging with this setup. So using that same DMD, we're going to do what's called holography. So what we're going to do is we're going to shoot a coherent plane wave through the sample, where they're going to re-interferent with a reference version of this plane wave. That generates a hologram, which is what I have over here. And what the raw data looks like is actually you have cells that imprint a phase shift into the light as the light travels through them. And so I'm not going to spend too much time on this other than to say this is something that really is a work in progress. But what we're really excited about is it can all be done on the same platform and interleaved at the same time. So we can actually do what's called label-free imaging, where you get these nice images of the refractive index, the density of your cell sample, and then you can track features. The thing is, you don't know which features are which, and then you can interleave that with a structured illumination to figure out which features they are you're looking for. And we think this is actually going to be the path forward to solving this cilia problem that I mentioned at the beginning. And this is just a picture of what that light path looks like. So this objective would both shoot the plane waves or the SIM patterns come out of this objective, the fluorescence is collected by this objective, but the holographic plane wave is collected by this objective and sent to the interferometer that lives above it. Okay, so just to summarize this section, and then I'll talk about the axial improvement we tried to do on the transfer function. What we were interested in doing was coming up with a way to do polychromatic control of light using a very fast amplitude modulator. We wanted to do that because we want to be able to try and track these biomolecules moving inside cells very quickly. So to do that, we looked at the way that DMDs work. We realized that there's some very, very nice diffraction physics that's occurring here that hadn't been solved analytically yet. Peter Brown, the postdoc in my lab, went through, found this beautiful analytical solution of how to figure out when the blaze condition is satisfied, which is where the diffraction orders you're interested in and the intensity envelope due to the configuration of the DMD overlap. That allowed us to build a microscope where we can exactly predict how the spatial frequencies are going to travel through the excitation arm, which allowed us to build a very nice way to do the structural illumination microscopy at sort of fields of view and speeds that have previously been inaccessible, particularly for multicolors. But none of this really got us to better axial transfer functions, right? And I showed you that in that blurry background of the data. So separately, we've been thinking about how do we get high contrast 3D imaging? So if we think about the way 3D images are typically formed in a wide field microscope, which is what the SIM is, we flood the sample with illumination light. Let's pretend we're not doing SIM, we're just putting light in. We then point the microscope objective at some plane, and maybe we move the sample up and down to generate a 3D image. Each of these little red dots here would be our fluorophores. When we're flooding the light with flooding the sample with laser light, we're lighting them all up. That's why we get background blur. In confocal microscopy, you focus the laser beam, and then you use what's called a confocal pinhole to reject this out of focus light, so you only see the fluorescence from this middle section. What we really want to do is combine the best of both worlds. We want to limit where the laser beam is shining, but then also take a picture. Why do we want to take a picture instead of this little region here? Well, to generate this entire 2D plane, we have to sweep this laser back and forth, which means, again, we're back into this, we're losing time resolution. So if we can somehow figure out how to meet these two things, then that would give us the kind of the best of both worlds. And this does exist. It's called light sheet microscopy. And in fact, the ideas that surround this have been known for a long time. So in 1961, Minsky proposed the confocal microscope. This is this patent. The way the confocal microscope works, again, is you shine illumination in, you then detect the fluorescence, and then you use a physical aperture to block any background fluorescence. So this is that strategy with the confocal microscope. But actually, roughly 60 years before that, Zsigmondy proposed something called the ultra microscope. So here there wasn't fluorescence, the imaging was dark field. And the idea is light was come in, it was condensed by a lens, and then you had an orthogonal detection pathway. And he looked at silver colloids with this, and this won him the Nobel Prize in the 1920s. And he was actually able to, in the dark field, he saw different colors for different size and shapes of these silver colloid particles. And what I really like about this picture I'm showing you here is actually recently someone managed to get an ultra microscope out of a museum and repeat Zsigmondy's experiment, but also having the matching electron microscopy of the nanoparticles. And they realized that Zsigmondy's original findings using this instrumentation about what the size and shape of these particles should be was almost dead on. And so this to me is really cool because they went and found this old piece of equipment. And so here we have a different way of reducing out of focus background light, because what we're doing is we're just bringing that light in from the side. The other thing I want to point out is that Mondi realized early on that to get two high resolution objectives near each other, you might have to cut one of them. And I'm going to come back to that point in a couple slides here. So this idea was reinvented / readapted for biology as light sheet fluorescence microscopy or selective plane illumination microscopy, or any other number of acronyms in 1993. And so the idea here is you have two objectives, you put your sample between them. If we pretend this box is our sample, each of these little hard spheres is our fluorescent molecule. The laser only lights up these ones. So we record an image of these, we don't see these, and we get 3D imaging this way without any background blur. Importantly, now our transfer function becomes a convolution of what our laser light is doing coming in and what our detection arm is doing. So if we think about the blue as our laser light, the green is our detection. Now, if we think about the point spread function, then all of a sudden the convolution of these two is much narrower in the axial direction. There's no free lunch here. The tighter you make the laser focus, the shorter your field of view because it's a Gaussian beam, which means it diverges faster as well. So this is, an amazing method. Sounds great. The problem is for us and the experiments we're interested in is that to do this, you have to bathe the sample in your objectives. What do I mean by that? To get two objectives close enough to a cell, they need to actually be in the liquid. And so what you've done is you've broken this barrier that existed between the equipment, the microscope equipment, and the sample, right? In the confocal microscope, we still have this barrier. And just to really reinforce that home, if we think about how this works, and this red line's our boundary, we have to break that boundary to put two of these microscope objectives together. And these cartoons come from my colleague, Andrew York. So we looked at the microscope, the light sheet literature. We've actually done a lot of work in this kind of SPIM area ourselves for large tissue imaging. But then eventually what we said, oh, you know what? There is another class of these methods that work. They're called oblique plane microscopes. So what does an oblique plane microscope do? An oblique plane microscope works on an idea called perfect remote focusing. So this pig is actually what people like to call an optical illusion, what I like to call really nice physics. So in this case, the pig is actually not there. What's happening is the pig is sitting inside two of these parabolic mirrors, which are satisfying a perfect imaging system. And so you're actually able to look at the pig from any directions up above, and you actually see a perfect recreation in any way that you look at it. So Botcherby, Wilson, and Booth realized that this can be exploited for imaging. So what does that mean? If you have a perfect imaging system, if we think about it in a purely geometric sense, all of the rays are put back together everywhere. So all of the rays that emanate from this object come back together perfectly, and they do that perfectly for every object. The problem is, for this to be satisfied, the magnification of your imaging system has to be a ratio of your refractive indices. Well, often in biological imaging, we're using sort of fairly high magnification, 60 to 100, or maybe even 20. That's definitely not true if you use an air detector, right? Your camera's in air, and then your sample's in water. This just doesn't work. So most microscopes instead only satisfy what's called the sign condition, which means all the rays come back together here in one lateral plane. And really what this comes to is you're satisfying Snell's law. This is the sign condition. If you want to have perfect remote focusing, you also have to satisfy the Herschel condition at the same time as the sign. The Herschel condition is the condition that anything along the optical axis is put back together correctly. So if you combine Herschel and sign, you get a perfect imaging condition. So how can you possibly do this for a biological sample? And realize the motivation here is we're looking for a way to make a 3D picture with only one objective. Well, if we think about a point emitter that emits light into a microscope, you can write down what's called the pupil function, which tells you the amplitude of the light that's coming out of the back of your microscope objective. In certain approximations, this is what the equation looks like. And so it tells you something about the X and the Y position of where you are versus the frequencies that are contained in the pupil function. Well, the nice thing about linear optics is it's reversible. So if we know what that pupil function is, we can actually just build a second microscope to just do the opposite of that pupil function. And now what we can do is all we have to do is satisfy the rules of perfect remote focus by having the ratio of these magnifications be the ratio of the refractive indices. And so here's what the two pupil function looks like. If we set that magnification so that it satisfies the sign condition, it turns out you satisfy perfect remote focusing. And so the rest of this math is worked out in this sort of seminal paper in this field by Botcherby, Wilson, and Booth, which is given here. So you could just imagine moving your camera to generate a 3D image, but actually what this does is it introduces spherical aberration. So here's if our native image plane, if we just shifted our camera back and forth, you get blurry point spread functions. If you do this perfect remote focusing, you now have a copy of that pig. So this would be like the optical illusion, the pig would live here. You then use a third microscope to reimage that. And actually now you can go 80 microns above and below. And importantly, this works at high numerical aperture. So actually you can do this in the single molecule regime. It's great. So all we have to do now is try and make a light sheet out of this. So here's our perfect remote focus system. We have our sample. We have that copy of our sample here. Here's what we see on the camera. Let's just tilt it. So if we make the laser beam come in at an angle, that would be a light sheet. And then if we just tilt this system, because remember, we can look at it from any direction, we'll get an image. Well, it's not quite that simple. High numerical aperture objectives have very short working distances, which means they like to collide with each other when you tilt them. So the oblique plane microscope that I mentioned, what Chris Dunsby proposed was, well, let's just lose some resolution. Let's use a lower resolution tertiary imaging system so our image gets blurrier. So now everything looks a bit bigger in the microscope because remember the transfer function is not as good anymore, but we can still satisfy all the rules of remote focus. And now we have a light sheet. So this was a method that has been used a lot. Elizabeth Hillman's group uses this as part of a method called SCAPE, where they do very fast imaging. Ben Yang, who's now at the Chan Zuckerberg Biohub, realized that if you just introduce a refractive index mismatch here, you can actually bend the light such that you can actually use a high numerical aperture objective again and retain the rest of your fluorescence. How he did that is he put a little wedge of water in front of this. So we tried to build this thing. It's really difficult to build. And so people look back and they look back at that old Zygmundi paper and they thought, okay, what we can do is something very similar. If we make an objective that has 0 working distance and we cut it, what we can do is we can exactly put that glass surface at the focal plane of that secondary objective. Then all the light that hits it will bend and go into the objective. And we can build a high numerical aperture remote focus system that acts as a light sheet. And so this objective's nickname is Snouty. And we've put a lot of work into building, understanding, modeling this system. And all of that is contained in this this joint eLife publication with Andrew's group and Redo's group. So this is what our version of the instrument looks like. So actually, this is a light sheet. There's a light sheet that comes out of here, but there's only one objective on it, which means we can do things like use fluidics. We then have a bunch of optics that let us do very fast, rapid scanning if we need to. And then here's what that cut objective plus the secondary objective looks like. Here's the results from characterizing the system. The main thing I want to draw to you here is that the resolution is about the same across the entire field of view. So it has a field of view of about 200 microns. It has a sort of resolution of, let's say, 300 microns before deconvolution in XY and roughly 700 microns in Z, depending on the light sheet. And importantly, it matches other high resolution 3D imaging methodologies. That includes spinning disk confocal microscopy and lattice light sheet microscopy. So here's the XY and then the XZ point spread functions for each of those types of microscopes. And so, we're really excited about this, and we've put it to some really cool uses that I just want to highlight before I end my talk. So the first thing we checked was, do we actually get a better transfer function in 3D? And the answer is, as you make that light sheet focus tighter and tighter, you go from that wide field situation where now what we're doing is we're looking at the Fourier transform of the average point spread function. So this is the transfer function again, but we're looking at the 2D version of it. And then now in this direction is Z, and in this direction is X. So we're looking at a cut. So you can see we start to get more information as Z as we use effectively tighter and tighter light sheets. So what that means is effectively a higher numerical aperture, which means the sheet focuses faster and defocuses faster. And in fact, we can get almost isotropic information if we start to really create a very short, tightly focused light sheet. The data that this thing takes is a bit confusing. It's tilted relative to the cover slip. So here we're sweeping a piece of tissue through it. We're doing this by moving the stage at a constant speed. Each of the dots represents an RNA molecule. So we thought a lot about how to analyze the data. And it turns out that if you can do this in the native coordinate system, so where this gray is here, here's our emitter, here's where our sampling is. It turns out for point molecules, you can actually fit all the data in this weird skewed system. And you can do this much faster. So we've written a package to do this on the graphics card that lives here at this, this localized PSF. And let's come back to that flagella example that I motivated this whole thing with. So remember I said we needed to track the thing turning in 3D. Well, Frankie's worked very hard with us on this system. So here's the 2D version of our data. Now we can turn it on our side. We're imaging at 20 volumes per second here, and hope everyone can appreciate that we can actually resolve the helical turn of this in 3D at 20 volumes a second of a nanoscale object. So this object's about a couple microns long, and then it's diffraction limited in the other one. And so that turn is sort of less than 700, or it's about 800 nanometers. So we actually can see that turn occurring and fit that data. So we're extremely excited about this and its future prospects. Okay, so the last thing I just want to mention briefly is we're applying this to this idea called spatial transcriptomics, which is essentially gene sequencing inside of tissue. And I ran a little bit long on my introductory explanation. So what I'm going to do is I'm just going to jump to this example we did during the COVID shutdown. So we were shipped lung tissue. And what we did is we were able to image thousands of cells in human lung tissue. And we were able to label it for the RNA for the putative receptor that SARS-CoV-2 enters the cells in, which is ACE2 and TMPRSS2. And then we were able to label this also for an antibody label that tells us an important cell inside the lung that the infection is thought. So we were able to image in roughly an afternoon, something around 16,000 cells. We could then compare this to our collaborators' work where they used a standard microscope, and over the course of a few weeks, they were only able to image about 100 cells. And all of us together working in a large collaboration, we published this paper where actually we looked at also the gene expression and from sequencing, and we were able to kind of figure out that there is some 10% of cells that are expressing, these important proteins, and these seem to be the cells that are most affected in the lung for those people that have had COVID-19. So this is just one kind of health relevant application that having these super fast 3D imaging allows you to do. And why the 3D is important here is it limits that background blur, right? Because we're imaging in human tissue, which is highly autofluorescent. Okay, so I'm going to skip this part and just say, why did I give you this really physics heavy talk on imaging? So in my opinion, there's been a turn to that localization precision is kind of everything that you need for super resolution in single molecule. But actually for this next generation set of biophysics problems, 3D contrast is really what matters. And in particular, being able to go really fast in 3D matters a lot because either you want to image a lot of your samples to get statistics, or what you're interested in looking at is not static, like the case in our cells and our moving flagella. So the people that did most of this work are, like I said, Peter Brown did all of the DMD work. For the RNA labeling work, this was a collaboration with Purusha Matata and Yoshiko Kobayashi at Duke. Lei Zhao and Roy Krutoff in my lab did the work there. And then Rizal Harardi and Frankie were involved in the flagella work. And then I mentioned the other collaborators for the bits along the way. And then obviously there's been some funding that's helped us out. So sorry if that ran a little bit long, but I'll stop there and I'm happy to take questions.
Thank you very much, Doug. So we could take some questions from the audience to start. And if you want to, go ahead and raise your hand in the chat if you want to ask a question. Do you want to come up here, Joel?
Hi, thanks for the talk. That was really, really interesting. I have a question about the holographic methods that you said were in progress. Are you able to do real-time hologram imaging?
So define real time.
Within a few seconds of the event happening, you're able to visualize it.
Yeah, so the hope is that we're going to be able to run at roughly, so we think we need about 8 or 10 images to do the full reconstruction in 3D for our approach. And the DMD can be can change its patterns at 9,000 patterns per second. So theoretically, we can go very, very, very fast.
Yeah.
And in fact, this is really important for us because it turns out that in the lab that we're in, our coherence actually drifts back around phase wraps on something on the order of like 10 seconds. And so we're fighting this, and we don't think it's the laser source because the bandwidth of the source is quite good. And so we think this is actually sort of either vibrations in the lab or something else occurring. And so really, we need to be able to sample on less than a second time scale anyway. So yeah, the point here is to go sort of at least kind of 500 volumes per second for the Cilia project is the hope. And so that we have multiple time points per going. So yeah, that's the reason to use the DMD for that. So
Cool. Yeah. Awesome.
Fantastic. Let's see. Looks like Jaime's got a question. Go ahead, Jaime.
Hello, am I clear? Can you hear me?
yeah, go ahead.
Okay, thank you for the talk. I really appreciate it. I thought your results were really cool. I did have a question when you were comparing your sim results with the others, and you mentioned how you had you're kind of skeptical of the deep learning results. I may have missed it, but I was wondering where that was coming from.
Yeah, so there's a few things that are going on in the sim community at the moment. So one of them is that people are trying to do an extra sort of processing step based on sparsity. And so they're just saying, look, fluorescence has to be positive. And then in that sense, I'm less skeptical of those results, although I have to wonder if they're not being influenced by noise. On the deep learning side, the reason I'm skeptical is that they are producing out of band results, so things the microscope didn't see, using less images for features that they didn't train the network on. And so what you see when you look closely into their data is you see a lot of periodic noise that matches the same type of reconstruction errors that I would expect if the noise wasn't reweighted properly. And so, you know, for larger macroscopic objects, I think they've got it fine, but for these objects that are right at the resolution limit, I question whether or not they're basically fitting noise when the network learns what it should be reconstructing. Does that make sense?
Yeah, makes a lot of sense. Thank you.
Oh, Miguel's got a question. So if you look in the, yes, Miguel, you can. Let me ask to,
Okay, thank you. So thank you very much for the seminar. Very, very interesting results, very thing. And I want to ask a general question. As you know, you've been using basically optical methods, but as you know, electron microscopy has more resolution, but it's always the problem of the vacuum that you need to have. But now there are these special stages in which you can put a live cell or a live sample in a liquid and see it in the electron microscope. So you will not get the same resolution that without the liquid, but still very good resolution. What do you think of this possibility? Think this is interesting or not? What
I think it's fantastic. And I think, you know, there's also been some very nice results on doing correlative light microscopy with the electron microscopy.
Okay.
And so I think for us, you know, there's a whole half of my lab that's biochemistry that I didn't really talk about, where we work on the labeling methods. And for us, this molecular identity of who we're looking at is really important. But for example, for the cilia problem, actually, we know what the cilia are, right? That there's only going to be cilia there. And so in that problem, you know, if we're not interested in necessarily which cargo is being transported, but just a transport problem, you know, you could start thinking about, you know, looking at that system with something with much better resolution and wouldn't have to go through the huge amount of effort that we have to go to even come close to approaching the resolution we need. So I'm really excited by what's going out by the electron microscopy community. And I think there's a huge opportunity there to start to mix and match the methods to move forward. Yeah.
Okay, well, thank you very much. Mm-hmm.
so we've got time for one more question. I think Blake had a question. Just give me one moment. Go ahead, Blake, and then after this, we'll have a Q&A with the grad students.
Okay, yeah, thanks again for the lecture. I had a question on your plane or light plane is what you call it. In the illustration, you kind of showed it at an angle going through the sample.
Yeah.
So my first question was, how do you determine like the proper angle to use, or the because the ultra microscope that you talked about from 1905 or whatever it was that at a perpendicular angle, or was that also at a skewed angle as well?
Yeah, so the ultra microscope, and in fact, let's go back just a little bit further here. All sort of standard light sheet microscopes typically tend to be at 90 degrees. And the reason for this is then what you're doing is you're looking exactly at the laser beam. And so you're forming an image of whatever is contained within the volume that you've excited. And so if we look at all these collections of things here, we see lots of right angles, right? So in each one of these, everything kind of looks like it is at a right angle, but the oblique plane microscope breaks that idea a little bit. So yeah, so how do you determine the right angle? So I would say we're all still arguing about it. And the reason I say that is it's ultimately related to the sort of resolution versus depth that you're interested in. So what I didn't mention is that as you tilt this little guy here over more and more, this thing we call snouty, this objective, you do lose a little bit of resolution in the tilt direction. And so if you back off the amount that you tilted, you now are actually seeing, you're launching your light sheet into your sample at a different angle. So you're actually seeing deeper into it, but you're actually not getting as much lateral resolution in the tilt direction. So in our case, the 30 degrees is about the critical angle for our microscope objective. It's just under it, which means that's the steepest we can bring a laser beam out without having TI with total internal reflection occur. So we went for that, which gives us the highest resolution, but gives us the shallowest Z depth. Other groups have argued that the thing they're interested in is further off the cover slip, so they don't tilt it as much, and then they shoot the laser beam, you know, at a different angle up into the sample. And so really it's determined by what your use case is, and then ultimately it's determined by the optics. You can't go steeper than that maximum alpha that I discussed that determines the resolution. You can't come in steeper than that.
Okay, yeah, that makes perfect sense. Thanks.
Fantastic. Why don't we thank Doug once again?